are added in, and they sort of nestle down around the larger bead with the DNA on it. And their purpose is that they're linked with one of two enzymes, sulfurylase and luciferase here, that affect the sequencing reaction, as I'll describe in just a moment. But they need to be down in there in the mix so that all of the reactions can take place, and so that the light can be produced when a base is incorporated. So let's look quickly at the sequencing by synthesis steps on the 454. We're going to imagine that we have one of our DNA capture beads here, and one of the millions of copies that are hanging off of it is going to be imagined right here. And this large gray blob is the DNA polymerase that is now seated at this annealed primer and ready to go. We then add in the first nucleotide, and this is a T. The first four nucleotides for this process are always the same, because these four nucleotides are always determined by the sequencing adapter. And as you'll see, there's 1A, 1C, 1T, 1G, and this is a so-called key sequence that now tells the downstream interpretation software what a single nucleotide incorporation looks like. Why is that important? Because what we're flowing across the surface of the flow cell are native nucleotides. So in the case where you run into like 4 A's here in a row together, all 4 of those A's are going to get incorporated against at once. There's no stopping A by A by A. They all 4 go together, and the downstream output of light is effectively 4 times as high as one nucleotide. You wouldn't know that if you didn't have that key sequence at the beginning for your software to look at and gauge all of the other incorporation cycles. So when this key gets incorporated, what happens? Well, we all know this. Basic polymerase biology, a pyrophosphate moiety is released. That goes through a series of downstream reactions that are catalyzed by these enzymes on the bead, and the output is essentially light. And that light is detected now by the CCD camera, which knows all the positions of all the wells that are emitting in these first 4 key sequences, and now records that cycle by cycle by cycle. Okay? And so we run these cycles effectively for several 100 times to generate the read lengths that are obtained from the sequencing instrument. I present for you here just sort of a trajectory of improvements, if you will, on the 454 instrumentation since this was introduced in 2005, where you can see that there have been increases in read length, so that with this latest Flex Plus, which we're just testing in our laboratory over the last couple of months, you can now get close to Sanger capillary read lengths actually out of this technology. So about 650 to 700, about 1 gigabase of data per run is being yielded, and this takes on the order of about 20 hours to complete. So it's an overnight run still. The error rate is about 1%, so you get 99% accuracy out of any given read. And we know that when you have an inaccuracy, it's typically in the range of an insertion-deletion type error. These typically are occurring now at those homopolymer runs like the 4A stretch that I pointed out to you, where that exceeds 6 or 7 nucleotides of the same identity in a row. You basically max out the detection on the CCD camera, and you can now no longer make that correlation back to the key sequence that I was talking about earlier. So that's a deficiency, but you can't actually typically make this up with what we call coverage, which means you don't just ever sequence through once. You actually have multiple molecules, which will include that homopolymer run, and the more you sequence it, the more sure that you're 6 versus 7 nucleotides of the same type, for example. So that's one way to get around the insertion-deletion error model here. The other advantage of this platform, which I think I pointed out here, this is a great platform for targeted validation where you're looking for single nucleotide changes, because the way the nucleotides are flowed one at a time, you almost never see a substitution error on this platform. The error rate for substitutions is extraordinarily low, and so if you're looking for a specific base in a PCR product or whatever, you can almost always detect that that's there and not be worried that you're getting some sort of a platform-specific error. Okay, let's shift gears now to the Illumina platform. This is around about the second platform introduced, originally marketed as Solexa. Do you have a question?
The previous technology,
yeah.
Single bead carries a single fragment.
That's been amplified.
Complementarity with a single segment?
That's right, yeah. So that provides the 3' hydroxyl for extension, if you will, and it goes and goes. Okay, so the Illumina technology, again, note the similarities now with what we've already been discussing. Library, which we usually are just for uniformity's sake, and you're good to go. So this is a very straightforward process. As I mentioned earlier, in our laboratory, because of the need to sequence thousands of samples at a time, in some cases for very directed sequencing projects, such as looking at case control cohorts, we've actually automated this to a very large extent to where we can produce on the order of about 1,000 Illumina libraries on a weekly basis, which is a technician and a fleet of small, inexpensive pipetting robots. So this is very automatable and works very well. In Illumina sequencing, the amplification of the library fragments now is occurring on the surface of a glass flow cell. So each of these technologies has their own nomenclature as well for the device that does the sequencing. In Illumina, it's the flow cell. And again, you can see sort of very shared characteristics here. The surface of the flow cell is decorated with the same type of adapters that are put onto the Illumina library fragments. This provides a point for hybridization of the single-stranded fragments. The amplification steps are essentially what's called a bridge amplification, where the DNA molecule will then bend over and encounter a complementary second end primer. and the polymerase essentially does multiple copies in one place, which results in this collection of fragments times several hundred million on the surface of the flow cell now, and this is called a cluster. So when you image a cluster during sequencing, it looks like this very bright little dot here, and when you image a portion or all of the flow cell, it begins to look like this star field, if you will, as the incorporated fragments are scanned with the laser. They emit a light frequency, and that's detected by the camera. That's coincident with the scanning by the laser. And I should point out that then there's another process that takes place when you go to the read 2 or the paired end read, which is essentially that you wash away all the fragments that you've already synthesized, you go through another round of some amplification, and now you change the chemistry for release of this fragment up for sequencing, and you effectively copy the other strand in the other direction from the way that it was first copied in this initial go-round. Okay, so how does the sequencing chemistry work? Now, this is fundamentally different than the 454 chemistry that I showed you in a couple of ways. First of all, we're supplying all 4 nucleotides into each step of the stepwise process. And in fact, the way these nucleotides are designed is very specific. So we have all 4 in the mix because each one, A, C, G, and T, has their own unique fluor, so they record at a specific wavelength when they're scanned by the laser. So that's why you can have them all in there at once, because you can get the identity back just based on the wavelength that's interpreted by the machine camera. In addition, at the 3' end, where normally you would have a hydroxyl available for the next base incorporation, there's a chemical block that's in place. And that chemical block doesn't allow you to incorporate another nucleotide until you go through the detection step and the deblocking step. That removes that and turns it into a hydroxyl ready for the next go-round of incorporation. In a subsequent step, the fluorescent group is also cleaved off because it has a labile connection here, and that also removes the fluorescence so that you don't get any background noise, if you will, for the next step of incorporation. Now, what I just said is absolutely not true 100% of the time, right? Because the one fundamental rule of life, if you don't walk out of here today with anything else, you have to understand that chemistry is never 100%, right? You probably learned that in college. And it's also true here. So 2 things can go wrong in particular. The block cannot be there, so it may not have been synthesized correctly. Some proportion of the molecules in this mix actually won't have a block, and you'll incorporate 2 nucleotides.